All right. Hello, everyone. Welcome back to the WebEVTalk series, a talk series for EV enthusiasts around the globe. So today is the 13th July 2021, and it's my pleasure to have Julie Gavard from CNRS Inserm, Nantes University in France. So Julie received her PhD from University Pierre Marie Curie in Paris for her work on cell-cell addition at the neuromuscular junction in 2004. She joined the NIH USA in Sylvia Gutkin lab and explored the molecular mechanism by which angiogenic factors modulate the endothelial barrier. And in 2008, she was appointed permanent researcher at CNRS France and focuses her research on the signaling mechanism and the intercellular communication between cancer cells and endothelial cells. So today, Julie is going to share with us her lecture, which titled Spitting Out the Demons, Extracellular Vesicles in Glioblastoma. So for this session, I'd like to thank Horiba Scientific, who has been kind to sponsor us through Patreon. So if you're interested to sponsor us, please visit the patreon.com and search for WebEVTalk, and you can actively sponsor us.
Okay, so thank you very much, Carolina. Thank you very much for the invitation. It's a pleasure to be here today at the EVTalk and discuss with you our research on the role of extracellular vesicles in brain tumors. This research is done in my team, signaling and oncogenesis, angiogenesis, and permeability. It's the SOAP team at the Cancer Research Center in Nantes. It's on the Atlantic shore, or nearby the Atlantic shore in France. So really briefly, the team is interested in deciphering vascular permeability in glioblastoma, which are highly aggressive tumors of the central nervous system with a poor prognosis, as the median survival range between 12 to 50 months. Current treatments include surgery when possible, followed by a concomitant radiotherapy and chemotherapy with the alkylating agent temozolomide. Despite this harsh treatment, harsh treatment, relapse is almost inevitable and fatal in a short window of time of 7 to 10 months. And therefore, second-line treatments are essentially palliative, such as the anti-angiogenic antibodies, such as bevacizumab, which targets the vascular endothelial growth factor VEGF. As in many other solid tumors, the loss of barrier integrity is a hallmark of aberrant tumor vasculature. And therefore, elucidating the mechanisms of permeability might give new insight on edema formation, angiogenesis, and invasion, which are instrumental for a glioblastoma progression. From a molecular and cellular standpoint, the endothelial barrier is formed by the organization of endothelial cells together and articulated around cell-cell junctions, with the tight junctions and notably Claudin 5, and adherent junctions with notably the VE cadherin. Another layer of complexity is added when you talk about the vasculature in the brain with the blood-brain barrier, as brain endothelial cells contribute to the neurovascular unit and are in interaction with astrocyte, neurons, and pericyte. These highly specialized brain endothelial cells are highly sealed and also express a specific repertoire of transporters. So one question we try to address in the lab is, what are the permeability factors involved in the loss of barrier integrity and their mode of action? So to do so, we first set up an ex vivo caricature model with GBM cells and brain endothelial cells. As for GBM cells, we decide to implement and work with patient-derived cells with stem-like properties, or GSC, for glioblastoma stem-like cells, because these cells recapitulate the tumor ontogeny from the tumor of origin. Basically, these cells can self-renew, they are multipotent, they induce tumor in plant and animals, and they're also resistant to conventional therapy. They also express some stemness markers such as nestin and SOX2 as seen here. They grow in 3D as spheroid, non-adherent, and in the absence of serum, which is quite convenient when you want to work with extracellular vesicles. On the other side, we culture normal human brain endothelial cells that recapitulate the properties of the brain endothelial cells within the blood-brain barrier. They organize nice and tight junctions, they form a barrier, and they still retain some plasticity as they can sprout and form tubule, and their permeability can be increased or decreased. So with this model, the first thing we notice is that these GC, patient-derived glioblastoma cells, modify the endothelial barrier morphology and properties, and indeed conditioned media from GSC in culture increase permeability of brain endothelial cells. And at that time, we identify few factors involved in this propermeability effect: the cytokine IL-8, which I won't talk about today, and two other cytokines, the vascular endothelial growth factor, VEGF, and the guidance molecule semaphorin 3A. And then we decide to explore more in depth their modus operandi and how the GSC release the propermeability agents. And one thing that we also notice when characterizing this GSC is that there are both a low lysosome flux and a high vesicular trafficking. So obviously, we went on and tried to characterize the extracellular vesicles from these cultured GSCs. And to do so, we used the classical differential ultracentrifugation procedures and characterized EV from cell culture supernatant quantitatively and qualitatively. Interestingly, as you can see in this picture here, EV produced by GSC are efficiently and quite rapidly uptaken by brain endothelial cells in culture. So then the next question was, what could be the functional impact of this glioblastoma EV on brain endothelial cells, at least in vitro? And as you can appreciate on this sprouting assay, extracellular vesicles from GBM cells induce brain endothelial cell sprouting and also increase permeability here in vitro and in vivo. The different numbers here correspond to different patients. We then move on and characterize the proteinangiogenic cargo of this GBM EV, and among the different factors, we notice the presence of the vascular endothelial growth factor A, VEGF. And indeed, this was confirmed with ELISA on EV from patient cells and also on EV isolated from plasma of newly diagnosed patients. So VEGF is transported in GBM EV. So to then address whether this EV or this VEGF within the EV is responsible for the EV effect on angiogenesis, we then first block the VEGF pathway in brain endothelial cells with classical VEGFR and tyrosine kinase inhibitor. And as you can see on this in vitro permeability assay, blocking the VEGFR in brain endothelial cells reduce the effect of EV on permeability here and here. We then go, we then pursue with a more molecular approach and knock down VEGF in the donor GSC cells and isolate EV in which VEGF-A was depleted, as you can see here with this ELISA. Again, when VEGF-A concentration was reduced in EV, the permeability effect of such EV was largely reduced, as and the same was true with sprouting assay, as you can see on this image, and further quantification. So this first set of data show that GSC, really VEGF in the classical secretory pathway and soluble, but also is transport within EV, and this VEGF is active and functional on brain endothelial cells. So what about the guidance molecule semaphorin 3A? Interestingly, again, with an ELISA, we found that in different patient-derived cells, extracellular vesicles, semaphorin 3A was detected by ELISA within this EV. And again, the same was true when measured in extracellular vesicles isolated from the plasma of newly diagnosed patients. We then deploy a similar strategy by knocking down semaphorin 3A from donor cells, isolated semaphorin 3A depleted EV, and applied to brain endothelial cells. First, the ELISA showed the depletion of semaphorin 3A in this EV, and then permeability was reduced in vitro, as you can see here, and also in vivo using a subcutaneous or a measure of the permeability, vascular permeability of the dermal blood vessels. So we wanted to check more this effect of extracellular vesicles in the context of the blood-brain barrier. And to do so, we revisit a pioneer experiment that were set up to highlight the presence of the blood-brain barrier. More precisely, what is done here is to inject an Evans blue dye systematically in animals, and when the blood-brain barrier is intact, the dye cannot extravasate and colonize the central nervous system tissue. But when there's a leak in the blood-brain barrier, then the dye can extravasate and be found in the brain. And actually, that's what you can see here with these images: intact barrier, no blue dye within the brain. And interestingly, when we apply extracellular vesicles on the meningia of the mouse brain, you could detect this blue extravasation in the brain on the left hemisphere where the drop was deposited, and also on the right hemisphere, supporting the idea of an active transport throughout circulation of the extracellular vesicles. Interestingly, when semaphorin 3A was knocked down in donor cells and depleted EV was applied, this leakage was less prominent. And this is also seen with these images here, and quantification in different territories of the brain with an increase of blood-brain barrier leakage with extracellular vesicles, which was reduced when semaphorin 3A concentration was reduced. And this was also seen to a lesser extent on the right hemisphere, again supporting this idea of an active transport. So with that, we decide to look at what could happen in an experimental model of glioblastoma, because in here, remember that these mice are tumor-free, so no tumor, they're just healthy animals. So what we did then was to implant human GSC either intracranially or subcutaneously. We let the tumor grow for 5 weeks and then extract the peripheral blood and isolate EV from circulating EVs. Interestingly, after much characterization, we found that following mouse CD63 in either non-tumor-bearing animals or xenograft animal, there was not so much difference in the content of mouse, so host extracellular vesicles. However, when we measure human CD63, so presumably CD63 positive extracellular vesicles from the tumor xenograft, we could detect a small population of human extracellular vesicles circulating in these animals, with much more concentration in the ectopic model than in the orthotopic model. This might be due to the tumor burden between xenograft and intracranial, or the accessibility to the peripheral circulation. Nonetheless, when we measure by ELISA human semaphorin 3A, so again, coming from the graft, the tumor graft, we could also again detect human semaphorin 3A in isolated extracellular vesicles in both settings, and again, much more concentrated in the ectopic subcutaneous model, but still detectable in the orthotopic intracranial model. So this was quite interesting to show with these two different models that both semaphorin 3A and VEGF-A were delivered through extracellular vesicles to endothelial cells. That's not the only route, because these two molecules are also really soluble in the extracellular medium. There are these EV transport propermeability agents are still capable of inducing vascular permeability, and also they can be disseminated throughout the body, at least in the experimental mouse models, although with the few patient plasma study we did, it suggests that it might be also the case. So of course, there's many questions remaining, and we're gonna go one by one to all four, maybe not the last one of this question. And although we don't have a definitive answer for all these points, I want to share some data on this specific hypothesis. So the first one is whether these extracellular vesicles can convey resistance and serve as a resistance marker in patient. So to do so, we went back to our in vitro model and patient-derived cells that are in vitro resistance to the chemotherapy, the standard of care, temolizumab, as you can see here, while this drug is reasonably toxic to their differentiated counterpart. And this is also seen here with these images where TMZ is basically, like these cells are basically insensitive to TMZ, temolizumab treatment. So then we wanted to know what could be the effect, the impact of chemotherapy treatment on extracellular vesicles released and produced by these GSCs. So this is shown here, the isolation process, and then some quantitative analysis. First, it looks like the mean size of these extracellular vesicles are not much change between no treatment or treatment. But interestingly, upon TMZ treatment, the concentration of extracellular vesicles was largely increased, while the cells were viable and there were no effect on proliferation or cell death. So we then asked whether TMZ was not only changing the amount of extracellular vesicles, but also their protein cargo. So to do so, we perform a mass spectrometry proteomic analysis of extracellular vesicles isolated for untreated or TMZ treated GSCs. And this highlights a core of common protein, most of them falling in the extracellular vesicle Goff function or vesicular trafficking Goff function. We then decide to focus on the proteins that were differential between untreated and treated cells. And this is what you can see here on this network analysis. And interestingly, two signatures, proteasomes and ribosome, was shared between the two, not same protein, but same go function. Interestingly, there was this third network highlight only upon treatment, which correspond to actin cytoskeleton cell adhesion migration invasion process. And this is something that excites us quite a bit, because it means that the protein cargo with possibly a protein involved in adhesion, migration, and maybe further invasion upon relapse might be also transport within this EV. So this is something we are currently addressing, is whether this adhesion migration profile is involved in tumor invasion and relapse upon treatment. So of course you understand here that we didn't have the answer yet on this, and hopefully for next talk. So having said that, we then decide to pursue our quest on what could be the role of extracellular vesicles in patient. And one thing that excites a lot of labs and is a top intense field of research is whether this EV and possibly their cargo can serve as prognosis markers. So for this, we took advantage of the existence of a longitudinal cohort within the hospital here in Nantes University. And just for those that are not fully aware about the GBM patient therapeutic regimen. Usually upon diagnosis, patient undergo a surgery, and then this is followed by a concomitant radiochemotherapy with the temolizumab treatment. And then they pursue the treatment with a second shot of TMZ chemotherapy. And then they follow up and they might have a second line treatment. And relapse is most of the time occurring during this period of time. So we were lucky enough to have around 10 patients with a liquid biopsy at different time point of this treatment. And with that, we proceed to the isolation of extracellular vesicles with size exclusion chromatography, optional differential ultracentrifugation, and did characterization by EM, expression marker inclusive or exclusive, and also monitor CD63 with ELISA. And here you can see a typical electron microscopy image from this isolated EV from patient. And here you can see the dynamic of this plasmatic EV concentration or vesicolemia in longitudinal analysis. And you can see that this concentration is not steady and is fluctuating and might correspond to different specific time point and milestones of the therapeutic treatment. So we decide to go in depth in all these steps. And the first thing we notice is that after the full radiochemotherapy treatment here, there was a drop in the EV concentration. This might be linked to also the reduction in the tumor burden, but also to the fact that somehow tumor cells are less active, let's say. Then one interesting fact is that when we measure vesicolemia at the beginning of the chemotherapy treatment and at the end, there was no much change. So chemotherapy was not increasing the number of EVs circulating in the body after this time of radiochemotherapy. So a bit different from what I show with the in vitro model, but this may have many explanations. Then we decide to look into more longitudinally, and as you can see here, before this recapitulate all the data with the 10 patient, reduction after the radiochemotherapy regimen, and then a slight increase here upon first line and second line treatment. Again, this might be linked to tumor burden. And actually, when then we clusterize patients before the one that undergo relapse and the one that either die before or had no relapse, we could see that at the end of the RCT treatment and upon relapse, there was a slight, but still reproducible increase in the extracellular vesicle concentration in the extracellular vesicle circulating in plasma from this patient. So I'm not saying here that we have a quantitative marker for a relapse, but a solid data suggesting that extracellular vesicles concentration dynamically reflect what might be going on with tumor progression. So again, we decide to go deeper in the protein content of these extracellular vesicles circulating in body fluids. And what we did was to, again, similar strategy, went on for a label-free mass spec analysis using 3 donors on one side and 3 GBM patient on the other, isolated EV by size exclusion chromatography and differential ultracentrifugation, and then this label-free mass spec. Again, this showed that most of the protein were from the EV core, which is quite a good sign of the procedure. And again, this was the highest goal function highlight with this analysis. Interestingly, the PCA principal component analysis showed different clustering between donor and GBM isolated EV. And again, we decide to focus on what was differential between the two populations with, as you can see here on this volcano plot, 2 proteins that seems statistically more present in the extracellular vesicles from GBM patient and some that were down-regulated. And among these 2 upregulated, we decide to focus on VEGF, the von Willebrand factors, because this was also associated with poor prognosis in patients using RNA-seq analysis. And as you can see, interestingly, when we measure with ELISA the concentration in extracellular VEGF from extracellular vesicles between healthy and glioblastoma patient, you see this increase in the concentration of the von Willebrand factor in EV isolated from such patient. And this is not shown here, but this was not the case when we measure extracellular vesicles in the total plasma and not in the EV fraction. So, of course, many questions still ongoing and active work on this topic, whether this VEGF concentration is modified upon tumor progression, because here is everything was done at the diagnosis time point. So again, we're going to take advantage of this longitudinal cohort and analyze the variation in von Willebrand factor concentration. And one intriguing question is, what is the source of this von Willebrand factor positive extracellular vesicles? And this might be super challenging, actually. So progressing again in our set of questions, one work that we are still ongoing in the lab is to try to understand the mechanisms for this constitutive extracellular vesicle production in the GBM cells and notably by this GSC population. So to start on this, we decide to interrogate the Cancer Genome Atlas, TCGA, for whether some let's say gene associated with extracellular vesicle was reported by many publications and reviews were kind of linked to the probability of survival in GBM patient. And this is reported here with around 50 different targets, and some of them were actually associated positively or negatively with a change in the probability of survival for GBM patients. And among those, you can see here that 3 were were also showing differential expression between LC and LC donor and GBM patient. And we were quite happy to see RAP27 among those, because this is the typical extracellular vesicle mediator, and this kind of validator strategy. And interestingly, we decide to look at MLKL, which kind of follow the same trend than RAP27. So MLKL is a pseudokinase which is typically involved in a certain form of programmed cell death, necroptosis. But it was also recently shown to be involved in constitutive extracellular vesicle constitutive or death-induced extracellular vesicle production. And so what we decide to do is to look at the effect of blocking MLKL on the release of extracellular vesicles using GSC cells. And as you can see here, when we block MLKL with a pharmacological agent called NSA or with a siRNA, in both cases, the EV concentration was reduced. So this shows and suggests that MLKL is involved in this EV production by GSCs in culture. So then we decide to check what could be the effect of blocking MLKL and therefore reducing EV release on the functional properties of GSCs. And in both cases, again, using the pharmacological compound NSA or siRNA, in both cases, the spheroid formation that reflects self-renewal abilities of the cells was reduced, as well as proliferation, as you can see here, with the EDU incorporation. So interestingly, blocking MLKL is linked to a reduction in EV, and this is accompanied by a reduction in the spheroid formation and proliferation of GSCs. So with that, this prompts us to look at what could be the impact in vivo. And what we did was to implant nude immunocompromised animals with GSCs intracranially, and then after the tumors were in place, start treatment with the NSA, the MLKL inhibitor, 3 times a week and follow tumor progression. And as you can see here on the endpoint, vehicle-treated animals show these massive tumors within the brain, which is largely reduced within the NSA treated animals. And this series recapitulate here over time. And this is also associated with a slight but significant survival advantage for the treated animals. Although we cannot formally show that this is linked to the role of EV, we nevertheless collect extracellular vesicles from the blood of such animals at the endpoint. And as you can see here, the NSA treatment was associated with a reduction in the EV circulation in this animal using human CD63 as a readout, so presumably linked to tumor burden again. So with that, I'm almost done with my presentation today. And so we show here that tumor-derived, GBM-derived extracellular vesicles alter the vascular barrier function. In addition, it looks like treatments, and notably temolizumab, alter the EV quantity and quality. What's the impact? Would be a follow-up, and this EV might contribute also to tumor progression. So right now we try to understand better what are the mechanisms linking this EV release with tumor expansion, and how do treatments alter EV quality, quantity, and are they paradigm to sustain tumor progression and relapse eventually. So with that, I will conclude and thank the team member, past, present, and future, working on EVs and helping on the GBM and signaling, GBM and signaling in the team. Also, all the core facilities that help us and our collaborators, many in France, but also in China, and our funding grants for this work. And thank you very much for your attention.
Thank you very much, Julie, for the presentations. There's a lot of data that you presented here. It's very amazing to learn about how the EVs play a role in the glioblastoma. So I'll invite everyone who wants to ask some questions. You can write down in the chat. And if you want to follow up someone question, you can just raise your hand. So I think I'll just start with the questions. So there's a few cargo or EV associated cargo that you shows here. Are they interrelated somehow? Are they belong to or influence similar pathways? Can you comment on that?
So, well, basically, with this approach, most of the time we decide to go to look at what's differential between, you know, treated, untreated, or healthy and patient. So obviously, this remove all the mediator and core component of the EV. So which might be shared and sometimes not, because with the mass spec, with the sensitivity, sometimes you might miss some. It looks like, but maybe this is the background of the team also, that adhesion molecules were quite important in the different settings, and this is something quite interesting because at some point you may want to see something that helps to stick EV to the membrane of the recipient cell. So adhesion molecules are quite present, and glycoprotein as well, which most of the time are also adhesion molecules.
Mm-hmm. Yeah, I have a question here from Esther, actually. Esther, would you like to ask, please?
Yeah, thank you very much, Julie, for this very interesting presentation. And I was actually wondering, you tested the effect on the permeability, the effect of the GBM vesicles on the permeability of brain endothelium cells. Did you also check endothelial cells from other tissues with some other specificities? And do you see the same effect, or is it really tissue specific or endothelium of this tissue specific effect?
So no, we decide to go for brain endothelial cells because, you know, this is the organ where most of the GBM cells will see first the brain endothelial cells. We also test like classical UVEC, which are more macrovascular and less differentiated cells. And we do see also this effect on permeability. We didn't, you know, explore that in depth. Also with a lot of the in vivo setting, this was done with dermal blood vessels. So it looks like this GBM EV was also doing such effect. So I guess the question then behind is, because these GBM EV are released throughout the body, whether we could detect some vascular permeability in other tissue. And this is actually was the first question of the oncologist when we isolated EV from patients. So this is something super difficult to address in vivo. Then one thing also that might be, I mean, this is also, we saw a lot of presentation and poster at the I7 on this topic, is that you also have some proteins involving hemostasis and thrombosis within this EV, and this is something I mean, this is something people are working on, and this might be also linked to the disease progression. So this we do see the von Willebrand factor. I know some labs have other proteins involving the thrombosis. So this is something we're gonna look at. And interestingly, this it looks like this. Marks are not associated with like the surgery, which is a quite strong vascular shock for a patient, but this is at distance, so it might be related to all these effects in. I mean, this might be interesting to follow up.
Okay, thanks. I think so too. Yeah.
So next questions. Is there anyone who want to ask questions? Otherwise, I'll go with my second questions.
Yeah.
So with the treatment-induced EV, are they majorly a good EV or bad EV? Are they sort of potentially carry also the drugs, for example? Have you studied that? Or
this is
could it be possibly a secretion process? Yeah.
Yeah, this is an interesting question. We didn't do that, and I cannot recall any other reports looking at that, but this is definitely because the cells we are using, they're really resistant. Most of the time it's because they're lacking the enzyme to to induce this DNA damage by the temolizumab. So but it might be also an efflux and multi-resistant drug transporter effect, which might end up using the EV. So for that, I cannot answer whether the drug is also efflux that way. What we saw upon treatment is really this emergence of adhesion and cytoskeleton link protein. This might be due to the fact that the EV genesis is somehow different, but that also be a way for the cell to expel some markers to further contribute to progression. So this is something we are currently addressing.
Is that also with other drugs, or is it just temolizumab so far with the actin abundance in the EV?
Well, I have to admit that we try only the temolizumab because that's the drug we use in patient, but this is definitely something that we can look at. Yeah.
Mm-hmm. Stefania, please go ahead. Okay, sorry. Okay, I'll just read this. Have you used any inhibitors to check the release of extracellular vesicles?
Can you read?
Have you used any inhibitors to check the release of extracellular vesicles?
Oh, inhibitor? Mm. Well, if there's any good inhibitor to block the release, I would be happy. Basically, I don't know if someone here can say there's an inhibitor to block EV release. We definitely, when we did all the work with the MLKL, SI, and NSA treatment, we also parallel that with RAB27 siRNA, and this was kind of matching the same effect. But we don't have a good drug. I don't know if someone has a good drug.
I think it's a bit tricky, yeah, because we know that there is a heterogeneous population of EVs, and it's well, there's a few different pathways has been reported. I think it's still a lot of discussion on what is the majority. Yeah. Yes,
sorry to interrupt.
You can go ahead.
Can you hear me?
Yes, go ahead.
Yeah, sorry, I'm having a bit trouble with my voice. I can't really talk loud. So because I've done some work with PAD inhibitors, so it's a peptidyl arginine inhibitors, and we actually looked at the EVs releasing glioblastoma. So I thought you might use some other type of inhibitors or other type of treatment, because we use as well tomolocytes. So that was the question behind. That's it.